that, I will turn it over to Mish, who will introduce today's speaker.
Thank you, Jackson.
Mhm.
So Tristan gave me a bit of a bio here, and I'll read that first. Tristan was born in a very outdoorsy family, um, and he was introduced to the natural world at a very young age. His initial interest was specific to South American birds, and subsequent education at Louisiana State University and the University of Kansas allowed him to accompany scientific expeditions to most South American countries, as well as China, the Philippines, Liberia, and Equatorial Guinea. It was obvious early on in his travels that Tristan much preferred those locations with less humidity, Western Peru, the Pampas of Argentina, and so on. And this was a significant reason he moved to Arizona in 2001. Tristan's botanical work is primarily centered around South American cacti, the best cacti, of course, and
Definitely.
the species of Ocotillo, Fucaria. So additionally, he continues to accompany scientific expeditions to locations around the world, hoping to again get to some desert-like locations. He currently lives in Chandler, Arizona, and is an adjunct researcher for the Biodiversity Institute at University of Kansas. He's a member of the Tucson Cactus and Succulent Society, an honorary lifetime member of the Central Arizona Cactus and Succulent Society, where he served on the board of directors. He currently administers their propagation education group and the seed depot. And he also volunteers at Desert Botanical Garden in Phoenix. What he left out of his bio is that he's been involved in the description of 3 new species of agave, 2 birds, and 1 cactus species, a species of Thelocactus, which I believe his last webinar was on. So on a personal front, I've spent dozens upon dozens of hours repotting seedlings with Tristan, and I've had the pleasure of working with him on conservation projects related to Brazilian cacti. And we've written an article together on the importance of maintaining collection metadata. And Tristan is one of the kindest, most genuine people that I know. He was one of the first people I met when I became interested in cactus conservation, and has kept me motivated through some good and some bad times. And I'm very proud to be his friend. I'm very proud to introduce him here today. Thank you.
All right, so with that, we will turn off our cameras and mute ourselves. If you have any problems, we will be here, but we are looking forward to your talk.
All right, well, that was a very nice introduction. Thank you, Mish. Yeah, let me share my screen here. And if somebody can confirm that they can see my screen.
Yep, it looks like it just came up.
Awesome. All right, well, thank you for having me as a speaker. It's always great to chat with you guys here at the CSSA. Today's talk is going to be kind of an extension of some research that Greg Starr and myself, and I'm sure everybody is familiar with Greg Starr, the Agavaceae expert here in Arizona. But we were doing some research in Mexico for several years and came up with a couple of different surprises. So this kind of talks about our latest surprise that we just completed the research on. So that's the title of our talk today, Agavaceae surprises in southern Mexico, but you can also call it, let's see here, I can't get my cursor to, there we go. Or research in Mexico, it's like a box of chocolates. You literally never know what you're gonna get. And this recent research is certainly indicative of that title. All right. So the start of this story for the most recent surprise was originally part of the result of a research we were doing on agave tenuifolia back in 2020 and 2021, where we had ended up finding out that what was formerly thought to be agave tenuifolia was actually composed of 2 different species, and the second species was undescribed, and so we did describe that in 2021 as agave cryptica. It had originally been confused with agave tenuifolia, and the distributions were clearly disjunct, and I put the distributions there. Agave critica is the one that's actually along the eastern slope in San Luis Potosí, Nuevo León, and Tamaulipas. And true tenuifolia is actually located in central Mexico, centered in Querétaro. Um, so when we were doing this research, part of what we did was we mapped out the distributions of the taxa to see, if there was a gap in the distributions, how they segregated, if they segregated, things like that. So we used a variety of resources and sources to determine the ranges. One of the primary resources we did use was iNaturalist, which many of you are probably familiar with. It's kind of a crowdsourced informational database on all living organisms. I mean, from plants to insects and birds and mammals and things like that, where you can go out there and you can post records of species on this database for everybody to look, and it will map it out on the world map. And so that's part of what we did in this analysis for Agave cryptica. But we were restricted to those pictures that included infructescences or inflorescences of the agave. So the blooming stalk or the fruiting stalk of the plants, because that's the primary indicator of which species was involved. So we only looked at records in iNaturalist that had photos of those components. So what we found is Agave cryptica is indeed segregated from true tenuifolia. Agave cryptica was the more northern populations and records, while Agave tenuifolia was primarily a central Mexican species at a little higher elevations. However, the interesting thing is in iNaturalist, we came across a couple of photos within iNaturalist that simply didn't match what we had been seeing with cryptica and tenuifolia. And they were identified as tenuifolia, but they were found way down here in Veracruz, which made absolutely zero sense from what we knew about cryptica and tenuifolia and our travels for the past couple of years in the course of the study regarding habitat and form and things like that. The pictures also looked kind of strange. So in our paper, when we described cryptica, we actually kind of punted. We didn't get to the actual locality of in Veracruz where these things were located. So we've simply added a statement within our paper that said records of plants assigned to the species in iNaturalist, well separated from other records of species, cannot be confirmed to belong to tenuifolia. So kind of punted it down the road, hoping at some point somebody, or maybe us, if we ever got down that area, might be able to solve that mystery of these weird plants down there. So to give you an idea of what those pictures were, these are pictures that came from iNaturalist. And like I said, they were identified as tenuifolia, but you can see the shape of the plant is very different from tenuifolia. The leaves are more blue-green than they are in tenuifolia. And it's got a very upright, erect, or what I call goblet shaped to the plant itself. And there were several photos of the plants out there, but the distinctive thing is they didn't have photos of flowers, but they did have photos of these infructescences here, which is infructescence is the old flowering stalk, but now bears fruit instead of flowers. And the main difference between cryptica and tenuifolia was that the tenuifolia flowers were well separated from each other, whereas in cryptica, they were bunched together. So in this photo, sure enough, the spacing of the flowers did match what tenuifolia flowers would do on the stalk. So it did look like infructescence-wise, it is a tenuifolia type of formation. But the plant certainly didn't look right. So that's why we punted and we kind of didn't know what to do with that set of records. So we left them as is. But Greg and I were continuing to do research, and much of our research is down in Oaxaca on agaves. And so we were doing a trip in October, November of 2021, where Greg wanted to get to all these different localities of these different species that he was working on, primarily for an additional book on agaves. And so we had planned on visiting areas in the south here Chiapas and also in Oaxaca here. So these sites were the sites we were going to visit. And when we were planning this trip, we were thinking, well, it would be nice if we could like come back the easy way, because there's a nice highway through Veracruz here, whereas all these roads down here in Chiapas and Veracruz has what we call topes, the speed bumps, and it was hell going through a lot of those areas. So it's like, why don't we take this fast road back here? Because right up here, where there's this 151, that's the locality of this agave tenuifolia suspect from iNaturalist. So it's like, yeah, we can knock that out and just see about what those plants are like when we're there. So that's what we decided to do. So we decided to make that detour. So we ended up going all to these Oaxaca and Chiapas localities for the agaves, but eventually we made it to that Veracruz locality. And the locality down here that I've squared out is actually one we added at the last second based upon what we found up in the Veracruz locality for tenuifolia. So this is the locality that iNaturalist led us to. There are GPS coordinates to the plants. And sometimes the GPS coordinates can be a little more generic than specific. But it was definitely the right area that we were in, these low hills near Jalapa and Veracruz. We got there fairly easily. There are roads that get pretty close to them. As you can see, there's some active mining because these hills are limestone hills. And so they were heavily being mined. We'd hear explosions all day and trucks driving by of the loads of the mountains. So the mountains are being taken apart basically. But this is the slope that we climbed up to get to these GPS coordinates. And it doesn't look steep, but trust me, it is steep. Greg is literally on his hands and knees for a reason, for good reason. The slope was quite vertical and we had to climb up on our hands and knees to try to get up the slope to the ridge. And then we were going to continue over the ridge to where we could hopefully find the plants according to the GPS coordinates. At the top of these ridges, it was beautiful. The clouds ended up coming in, so we were kind of sitting on top of the clouds. We didn't make it to the top of the ridge. So we walked along the ridge, and then according to the GPS coordinates, we started moving down the ridge through the underbrush, and finally we did find the plant. So here is that plant, that very same, very erect growth look to it. This picture is a little deceiving. The plants are a little more bluish green than solid green here. But once again, we saw these plants and found them and were able to touch them and feel the leaves. 100%, this is not agave tenuifolia, not even closely related to it as far as we were concerned. Obviously, Greg's the agave guy. He knows pretty much all the agaves, and we were both pretty confident this is definitely not an agave. So the question is, what is it? The leaves were very rough. The leaves actually had a furrow in them, like the leaves were curled up. So this sulcus was present in the leaf shape. But you looked at the plants, and in this picture, it's kind of hard to see. The plants themselves are kind of a blue-green here. The rest of the plants and vegetation, this is actually a species of grass. So it blends in very, very well with the grass in the area. Just a slight color difference, more blue-green instead of a brighter solid green. But the infrotescences on this plant, and here's some more individuals showing that classic goblet erect shape that's so distinctive and so unlike any of the agave, tenuifolia, or cryptica, or any in that group. The infrotescences themselves, once again, showed very separated flowers. We had no flowers there, just the fruits, but the fruits obviously came from the flowers, so we knew the separation of the flowers when it was flowering. But now that seeing him in real life, Greg was just astounded that the fruits on these things were so big. So that was another thing that clearly separated them from the agave tenuifolia group. The fruits were much bigger than those plants. So we knew for sure this was something weird. This was something different. The fact that we didn't know what it is. It was clearly an Agavaceae because it had the classic Agavaceae flowering stalks. But we didn't see any flowers. The infrotescences that we did find, as you can see in this photo, were extremely old, probably at least 2 years old. Most of the fruity capsules were rotting. So it was like, this is a mystery. What could these things be? So So since we knew it was Agavaceae, when we finally got down from the mountain and got access to the internet, immediately I started doing some research and to find out what these things are. My first place to go to when you have a question about Agavaceae for us is to agavaceae.com. And the nice thing about agavaceae.com, it has literature links, plus it has a list of all of the taxa involved in Agavaceae. Of course, this was before the additional genera of agave were described. So there's now para-agave and paleo-agave, echino-agave, but those are all included in the genus agave in their list currently. But I figured, okay, let's go through this process methodically and from top to bottom, let's go through each genus and see where does this guy fit? Because it doesn't even seem to be an agave. And so looking through the agave list and all the species possible and through Greg's extensive, knowledge about agaves, we were pretty sure if it's an agave, it's definitely new and incredibly distinctive if it's indeed an agave. So we moved on to the next one on the list, Bechernaria. Most people are not aware of Bechernaria. It's not something that's very common. Bechernaria consists of about 7 species, depending on your taxonomy. Almost completely a Mexican endemic genus. One species does reach outside of Mexico. Very poorly known and studied. Nothing in recent years really has been published on these plants. They have very distinctive pendant flowers, and mostly the leaves are rather wide and soft, and some of the plants can get fairly big. The habitat is usually humid and montane environments. We had actually run into Bechernaria up in northern Mexico when we were doing the research on the Agave cryptica, but they had very wide leaves and very distinctive shapes. But as in all things in Mother Nature, one species is an exception. That one species is Bechernaria calcicola. And it was described in 1986. This is the article. These are the photos from that article. And it shows a plant that is found in a rather arid environment similar to what we had. Calcicola is indicative, the name is indicative. It was also similar to habitat. So it's limestone loving type of habitat. The plants were similar in the pictures here. Didn't quite have that goblet erect shape. The leaves tended to be more lax. Of course, that's just a couple of photos, right? Flowers, extremely distinctive and classic Bechernaria, very pendant hanging flowers, well reflexed at the tips, beautiful red flowers in this species. And photos of the infructescence with the fruits here matched what we were seeing at this locality in Veracruz. So when I did get back to the States and I did start doing additional research, what I did find out, oh, I forgot about this. I'm jumping ahead of myself a little bit. After we found this and we noted this is probably a Bechernaria after looking at the literature in town, it's like, well, on the way back to Oaxaca where we were headed, we're sort of passing by the type locality based on literature of calcicola. So while the plant we had in Veracruz was fresh in our mind, let's go see if we can find those plants. And so we did. We took a side trip to the type locality of calcicola, and sure enough, we found the plants. The plants were very similar to what we were seeing in Veracruz. The leaves, the color of the leaves was more of a bright green, a dark green, a solid green, than they were a blue-grayish green in the Veracruz plants, but otherwise were similar. Plant shape, these were a little more lax, and the leaves were not quite as distinctively erect as the ones in Veracruz, but they were similar in that they were rough leaves and they did have that distinctive groove in them. And although we didn't find any plants in flower at the calcicola locality, you can certainly see here the infructescences indicating where the flowers were, which spacing wise seemed to match what we saw in the Veracruz plant. So it's like, cool, Bechernaria calcicola. That's pretty interesting. Although the ones we did see, we did, see these differences, but, we didn't see flowers on the plant. So flowers are usually pretty indicative within the Agavaceae of relationships. So when I did get back to town, did additional research, pulled up this paper that was from 1998 that sure enough said Beschneria calcicula, new report from Veracruz, Mexico. And it's like, aha. So looking at that paper, not only was it reporting Beschneria calcicula from Veracruz, but it was literally the exact population we had actually run into. The exact locality, the exact hill where we had gone and found these plants was where they reported Beschneria calcicula. So we're thinking, okay, then these plants are Beschneria calcicula. However, the interesting thing is in this article, it did not talk about flowers. The closest it had to mention flowers was inflorescence pink. Now, inflorescence can mean a couple of different things. It can mean the flowers themselves, but it often refers to simply the flower stalk, the actual stalk of or rachis of the inflorescence and not necessarily flowers. And, as you may guess, flowers are fairly important for telling different species apart in the Agavaceae, even beyond Agavaceae. So we were a little um, reluctant to jump on the calcicula bandwagon because we had started documenting several different differences at this point, including that goblet shape, especially on younger growth, the more bluish leaves, and the distribution, which was quite a bit farther north than the records from Oaxaca and Puebla that were included in the original description of Beschneria calcicula. We had also found differences in seed size. There were actually a couple of seeds that we had on the plants that were rotting at the Veracruz locality. So we were starting to see quite a few differences. We looked at details of leaves, and we found some differences in the leaf shape. So we were like, would really like to feel comfortable by having the actual flowers on these plants to really make a definitive decision about calcicula, because there simply was not enough information in this paper. So what about those flowers? So to accommodate that issue, we actually had another trip for plants planned for 2022 to get specimens of this, at the time, undescribed species of agave that Greg was working on, which we eventually described in 2022 as Agave salomonii. We went back to this place at the time when almost all Agavaceae were flowering. So we also figured, well, that's a good time to go back to this Veracruz population, because since all the other Agavaceae were flowering, surely this would be flowering too. However, when we finally did get to the Veracruz locality in 2022, we found the plants easily enough, but once again, zero indication of fresh flowering within the year at all. Still, it seemed like the inflorescences were at least a year, if not 2 years old. Nothing from the current year. So that gave us pause and gave us a couple of good pieces of information that we really needed. We need flowers to really look at this. We really had been getting on the bandwagon that this might be something different, but without flowers, we don't know how different, right? It could be a subspecies or a variety or something like that. It certainly seemed to have consistent differences, but flowers were critical. So the problem is that it seems the flowering times or flowering cycles for this taxon didn't seem to be regular, nor were they in sync with other Agavaceae in the area. The location makes it rather difficult to visit regularly because I live in Arizona and this is Veracruz, Mexico. So quite a bit of an effort to actually visit on a regular basis to check for flowering. And then in Agavaceae, once flowering cycle begins, flowering can often be relatively fast. So we'd have to have a very small reaction time for these plants if we were to catch them flowering. So how are we going to solve this? Well, Tucson Cactus and Succulent Society to the rescue. They have a research grant program that is continuous throughout the years. You can submit your research grant proposals to them, and they were able to provide us with a research grant to try to figure out when the flowering cycle for these plants is so that we can get down there. So what this grant did allow us to do is to provide for our Mexican botanist that we work with in southern Mexico, Gonzalez Garcia, for him to travel from Oaxaca to Veracruz. He would work with the community for permission to collect the specimens, and also work with hiring a local from the local village, which was not far away, to check on the plant progress throughout the year on a regular basis, 2 to 4 week intervals. Each time they would check, they would send us a WhatsApp message, because everybody has WhatsApp down there, and send us a photo of the plants to kind of see where they are in progress. And once they did start to show some kind of progress for flowering, we could hurry up and make arrangements to travel to Veracruz and get collection of flowering samples to coincide with those flowering cycles. So that was the plan. So Gonzalo went ahead and made the arrangements. So in June 2023, Gonzalo traveled from Oaxaca City to this locality in Veracruz to verify the location of the plants, and to work with the local community about possibly tracking the progress of these plants. And when he found the plants, I was very anxious to sitting by my WhatsApp waiting for him to let me know what he found. And this is what he sent. And I'm going to play this recording. It's actually a recording.
Mira, Tristan, sorpresota. Está genial la planta. Está súper chida.
So basically what he's saying there is this: look at this. What a surprise. This is really cool. He happened to be there and they were flowering. And so he sent a bunch of photos back of plants that were flowering, and seeing the flower here, clearly this is not the same as Bessernaria calcicula. Remember, the calcicula flowers were clearly a solid red, pinkish color. This is very different from calcicula. So at this point, we knew we had something new on our hands, and we really wanted to get enough information together so we could describe this new population. So it was very exciting. However, you'll notice the flowers in this photo are rather closed. They're not open. And what we found is we were late in the season. So the flowering had actually passed for that season. And most of the plants they found, and they found quite a few that had flowering stalks from that year, which was amazing since we had not found any recent flowering stalks in our previous trips. He found that most of them were starting to form fruits as opposed to flowers. Flowers were past. So unripe fruits, and they found, like I said, plenty of plants. You can see clearly the flowers are past their prime. They are actually starting to dry up and yellow, and that was pretty consistent across the board. Now, the inflorescence, the stalk itself, was indeed pink, so did match what the 1998 paper had indicated when they reported calcicula from this locality. So likely they had only seen plants that were not in full flower and didn't mention flowers, didn't realize the flowers were actually green and not red, and did not match calcicula. And instead they just mentioned the inflorescent stalk, which is indeed pink in both species. So a little frustrating, because we knew that we weren't there, or Gonzalo wasn't there at the right perfect time for collecting fresh flowers. You can see that once the flowers do complete anthesis, they actually start to yellow even. So we knew we were a little too late to get good specimens of flowers to incorporate into a description of this new taxon. So we need fresh flowers. That has to happen. We are past the season already for it, so it would have to wait for another year. So this is going on for like 3 years now. So to the rescue, once again, TCSS, the Tucson Cactus and Succulent Society, provided us the means by granting another research grant to us in March of 2024, which allowed us to travel back to Veracruz in 2024. And the proper, what we were hoping was the proper flowering season, working with the community to collect specimens with their permission, collect fresh flowering material at full anthesis, but also to revisit the type of locality of calcicula for comparative material, because all of the specimens that were available were very old specimens back from when it was originally described. Many of the specimens were actually sterile specimens, meaning they didn't include flowers or flowering components. It was just leaves and roots and things like that. So since flowers are the best way to tell these two taxa apart, we really would like to have recent specimens from the type locality. Now, Calcicula was also reported from Puebla and other localities in Oaxaca, so we wanted to also visit those localities, determine if there was any variation within those. Many of those localities were recorded as Calcicula based on sterile specimens, once again, so non-flowering individuals. And then we also wanted to visit similar locations in Veracruz and between the localities of our new taxon and Calcicula to determine where there's a break. Is there a gap in the distribution? Is there overlap in the distribution between the two species? Or is it a gradation or something like that? So all of those components were part of our next trip that we made in April of May 2024. So first order of business is we returned to the type locality of Bechernaria calcicula, expecting to find these beautiful inflorescences, the right time flowering with these red flowers. Finding the plants was not a problem, very easy. However, we did find quite a few plants that were in stages of flowering from what we could tell, because 100% of the flowering stalks that we found were eaten by goats. And it's hard to see in these pictures, but all you see is the stub of the inflorescences and the remnants from what the goat had actually eaten. This is a fairly common area for goat grazing for the locals. And we searched and searched and searched and found many plants that had fresh stalks from that year, but 100% of them were indeed chewed to the stubs. Very disappointing. So it's like, well, at least there are other localities for Bechernaria calcicula from the literature that we can go check, and hopefully they don't have this type of local problem with the flowering. The problem is the localities for Bechernaria calcicula were reported upwards of 40 years ago from when it was originally described in the 80s. And what we found when we started visiting those localities is 40 years has done quite a job on these localities. And most of the localities were completely changed and no longer harbored appropriate habitat, or everything has just been ripped out. So we found some localities are now completely agricultural. Everything's been ripped out. It's just agricultural. We found some where it's probably road construction went right through the area and destroyed everything. But we even found one locality that we found where the plants would be. We had almost GPS coordinates to the plants, the original plant localities. But 40 years has significantly changed the habitat, and it's now a pine forest. And there's heavy ground litter, heavy leaf litter, and it's unlikely that the Bechernaria calcicula existed in that locality anymore because it can't grow in that type of environment. And at some point, it must have been extirpated from that area based upon natural changes in the habitat. So 40 years did make a significant difference. But finally, we were able to find a locality where we found plants. But if you look at this plant, we started seeing these plants and they all had this goblet or rect shape. So we were going, well, this is weird. That was one of the differences between the new one and calcicula. So maybe that's not such a significant difference. And calcicula does have populations with that shape as well. So we're kind of bummed at that, right up until we found flowering specimens. And here's the first flowering specimen we found. And if you notice along the inflorescence for this plant, the flowers are indeed green, not red, which forced us to rethink everything once again. It's like, whoa, okay, so apparently what must have happened is these original populations of Bechernaria that were identified as calcicula were done so based upon sterile specimens, so non-flowering specimens. And because without flowers, they do seem very similar to calcicula, they were assumed to be calcicula. But now looking at the flowers, we know these are not actually referable to Calcicula. They're actually referable to the same plant that we found in Veracruz. So this was a revelation. So it made it even more important to find other populations that were flowering to ensure we knew what was the new populations versus the true Calcicula. Unfortunately, we didn't find many more. But what we did find was a couple of localities around this place right here, and they were all of the new population, the green flowered population. So our next job was to go back up to the locality we had originally found them, this new population, and see if we could get flowering specimens at that point. So hiking back up the hills, sure enough, for the first time ever, we saw signs of fresh flowering plants at some stage. And ironically, we saw a lot of them when we started looking that were in early stages, so we were too early for it. But we also found plants from later in their flowering process, so they're already fruiting. And for the first 45 minutes, we kept finding plants in these conditions, which was very frustrating. Just like we need one plant that has flowers at full anthesis. But finally, we did come across a plant in flowering, and sure enough, it matched the plants that we had seen at that other locality. And they were clearly green flowers. They were at full anthesis and matched the flowers that Gonzalo had found in flowering plants the year before perfectly. And so we were quite happy. We did end up finding additional plants, and they all matched. Green flowers, fairly well reflects petals at anthesis, very different and distinctive from Calcicula. So we knew at this point we did have an undescribed species on our hands, and we started to go about getting all the documented specimens and things like that, and collecting data from numerous plants that we found. So we're all one big happy family after having success in finding flowering plants of this plant that has frustrated us for years. Obviously, that's Greg Starr on the left, and everybody recognizes his smile. That's Gonzalo, our Mexican botanist, researcher, and myself, of course, in the front there, all happy with the new Beschorneria in the background. So what about Beschorneria calcicula? We still were frustrated because we didn't have fresh material from Calcicula. We really wanted to have fresh material to compare to fresh material. So what we thought about is like, well, we didn't spend as much time finding plants as we'd hoped because a lot of the habitats have been changed. So we had an extra day before we had to be back in Oaxaca and leave. So it's like, why don't we try the type locality again? And this time, maybe push farther into the brush, farther away from the road, farther up and down the road, and see if we can find somewhere a flowering plant that hasn't been chewed on by goats. The plant is very localized. It only grows on certain types of these limestone outcrops. So it's not like it was continuously distributed. So you're hoping to find other populations. So one more try, we headed back to the type locality, and boom, we lucked out. Actually, not too far off the road, Gonzalo started yelling from the car, "Stop, stop, stop. We got flowers." And sure enough, we had flowering plants that had not been damaged by goats. And looking at the flowers, these were clearly Calcicula, which we expected, but it was nice to have confirmation that sure enough, the flowers are this pinkish red color, very distinctive and different from the new population that we were going to be describing. And now we had fresh material for the comparative analysis. We did end up finding actually several more flowering individuals, and they all matched. Calcicula, which was expected. But it's always good to have confirmation. So we continued to find plants. We took an enormous amount of data on the measurements of the plant, not just the flowers. And there's Gonzalo's up on that little limestone ledge, yelling out measurements to me while I wrote them down on the nice comfy area where I could sit down. So that worked out really well for me. And at the end of the trip, we were all quite pleased, and we had another success photo with the usual three, but this time Javier, who was our guide and driver the whole time for all of these different years. He's a guide and driver that we use for all of our trips down there. He wanted it on the success photo, so we certainly were welcoming him because it couldn't have been done without him as well. So many thanks to Javier as well. So we were very happy campers at that point. So now, next steps, what do we do? Well, we got to take it back. We got to, compile all the data, and then we have to write it up. And a lot of people don't realize how much work these new species descriptions really are, especially in this case where it's not a brand new population that was, in some secluded valley somewhere. No, the history of this population was that it was actually misidentified as something else at first. And so there's a reason why that happened. And so you have to go into that and explain why we feel this is a population that actually deserves a separate name. So you got your abstract, your introduction, the history of calcicola and this population, our methods, results, the diagnosis, which is where you explain the differences between it and other species or among other species, what the relationships taxonomically are to other species as much as you could possibly imagine, as well as information about the ecology, conservation, like an IUCN status for the species to help in conservation and preservation, as well as an outline of the distribution. Because when we got back and we started mapping out the distributions of what we knew was the new one versus what we knew was calcicola and confirmed with flowers, we could also make some assumptions about all these other older localities that sometimes just didn't exist anymore. So here's the map that we put together of the two taxa. And green is the new taxa. So this is what we're calling the type locality where we actually found the plant in Veracruz. You'll see the border with Puebla is right there. So it's very close to the Puebla border. But these localities are also, the squares are where we confirmed 100% that the plants were that taxon. Circles is what we assume is that taxon. So we either found plants with no flowers or they're historical localities claiming to be calcicola that we now suspect are this new taxon based upon proximity to known populations down here, the square. So you'll notice these guys are north of Tehuacan and this wide swath of the Tehuacan dry valley. However, the calcicola plants that we knew and confirmed were calcicola were the squares, and the circles were nearby ones that we weren't able to confirm with flowers, but we made the assumption based on proximity to known flowering plants that those were indeed calcicola as well. So let's see. Green is the new taxon, and red is true calcicola. And you'll see this wide swath in the middle that separates the two. So they probably do not ever come into contact based upon this natural geographic barrier to gene flow between the two populations. So once we finished with all this, we put it together fairly quickly. We submitted it to Hazeltonia, which is, as you know, the publication, the peer review publication published by the Cactus and Succulent Society. We published it there for a couple of reasons. Obviously, it's well respected, but we also wanted to be peer reviewed because Veterinaria hasn't been dealt with in years. So we wanted to make sure that there were other experts that were reviewing this before it was published. Unfortunately, that ended up being kind of a problem because there weren't many people that knew about Veterinaria. So there weren't many people that felt they were qualified to actually review our paper. So Peter Breslin, the editor, was able to get one reviewer to review it fairly quickly, and they came back, I think it was within a week, saying, yeah, this is great, this is awesome, publish away. But we needed 2 reviewers. So we kept looking for more reviewers, and finally, I think it was November, we found another reviewer. They reviewed it. They gave it the thumbs up in, I think, early December. And one of the reasons we wanted to submit to Hazeltonia also is that it was now electronically published. As papers were published, it didn't wait till the printed version at the end of the year. And with new species descriptions, you're always in a race to publish the name. Because if you don't publish it first, whoever publishes the name first, regardless of how extensive the research was associated with it, that's the one that legally sticks according to the International Code of Botanical Nomenclature. So it's always worried. You always have a worry in your mind that maybe somebody else is looking at Veterinaria and maybe somebody else has a paper in press or they're working on a research project. So you kind of want to be the first to get that publication out there, and electronically nowadays is the best and fastest way to do that. So that was one of the reasons we were trying to get this done as quickly as possible. Now, we were thinking we're pretty safe. Beshrenaria really hasn't been studied in almost 30 years. So we just happen to be the only people working on these guys. Surely there's no chance of anybody else publishing on Beshrenaria right now. That would be way too hella much of a coincidence, right? Yeah. In late December of 2024, I was actually on vacation, Christmas vacation, Christmas break at my brother's in Seattle. And like any good botany nerd, I'm running around on my phone and on the internet looking for articles on Beshrenaria. And lo and behold, I pull up a paper that was published electronically just 2 weeks before that describes a new Beshrenaria from, wait for it, Veracruz, Mexico. And it was published 2 weeks prior in early December. My heart sank. It literally was almost a heart attack. And it's like, we have good data that we could add to this paper, but as far as, the naming of it, obviously that wouldn't be something we could do. So we'd have to retract the paper, revise it, and resubmit as just an FYI, just an informational type of thing on this new taxa. However, once I started reading through the paper, I did realize it happened to be a completely different species of Beshrenaria. It just so happened to be also in Veracruz. So all the little Swiss cheese holes started to align. It is actually found in this area on the other side of the mountain from the new one we had just described, on this area up into Veracruz, which is more higher elevation, montane, and it was pine forest. Description and pictures included in the paper showed that the plant was very different from what we had. It was a wide-leaved plant. It did have green flowers, but it was a very wide-leaved lax plant in a much wetter environment. So my heart subsided and felt a little bit better about it. And so finally, the paper was published electronically in January of 2025. So it got out there. We were all hugely relieved. But you may be wondering, well, what was that name? What did we name it as? And anybody who's worked in new species descriptions, there's always, a lot of discussion about what can we name it? What do we want to make it, related to the habit of the plant, or the color of the plant, or the location of the plant. Some people still give me grief about the Thelocactus Misha mentioned that I described a few years ago, because the species name I used was Tepomamensis, which you can't say 10 times fast. But that was as a favor to the community that asked us to name it after the community, and their community happened to be named Tepomeme. So can't blame me for that. But so I didn't want to make it too difficult, but we could name it after people. Anybody in the bird community knows that's a big, huge controversy nowadays, patronyms and naming things after people. I don't have a problem with it, but a lot of people in the bird community are reassessing that whole shtick. It hasn't gone into the botanical world, thankfully, yet. But or we could name it after something intrinsic about its ecology, where it's found, the type of substrate, even what pollinators there were. And that last one intrigued me. We because pollination syndromes may be different based upon flower colors. We were thinking, and we postulated in the paper that there may be differences in pollination. And we did see a moth on one of the green flowered of the new taxon, and hummingbirds were all over the place in both localities. So we thought that might be interesting. So what we did is we decided to name it after the most frequent and obvious pollinator that we saw in the field for this taxon, which he's pictured here. And that pollinator is Greg Starr himself. The goofball seemed to be very interested in demonstrating pollination, so I went ahead and named it after Greg Starr himself. So the new plant is named Beshrenaria starrii. Its close relative is Beshrenaria calcicola, but it is quite distinct and different. So as Greg will be seen now walking around, have you seen his Beschorneria? Have you seen Greg's Beschorneria? Because he certainly has. Not many of us have after, beyond that, but at least he has. So with that, I want to thank you. That's the story of Beschorneria starrii. Like I said, it just finished in January 2025. So this is pretty fresh out the door. But I do want to thank once again the Tucson Cactus and Succulent Society, without whose assistance this clearly multi-year project would never have been available to us or ever come to fruition. So, many thanks to them. So thank you, CACS, for having me, or CSSA for having me. And if anybody has any questions, I'm certainly will try to answer them.
Well, that was fantastic. I remember your Thelocactus talk, and it's always interesting to see kind of the play-by-play or the story of how some of these species get discovered, described, named. And so it was another fascinating view
Yeah.
into that world. I think I saw one question in the chat. I had a couple questions myself, so I'm going to be selfish and ask those first.
All right.
The first is that I noticed in some of your pictures of the green flowers of Beschorneria starrii, some of them look glabrous and others tend to look pubescent, especially kind of on maybe the ovary area. I was wondering if that had any impact, or I noticed it is kind of strange.
It's actually the hairiness, the amount of glabrous or hairy or hirsute growth on the flowers and the ovaries, as you mentioned, is a characteristic that is often used in distinguishing the different Beschorneria. So some of them do have it, some don't. Calcicula is described as having that, but starrii, it certainly seems like on average, I mean, there's definitely a variation in the amount of hirsute you get in the flowers, as you could see in the photos. But starrii did seem to have more typically than calcicula, but not enough to really make that a character that can distinguish between the two. It seemed like there are several taxa in Beschorneria that do have that character as well, and a couple that have are completely smooth. So it's an interesting character. I don't know how useful taxonomically it is in a specific scenario like this, but kind of a generic kind of separation of different types of Beschornerium, it seems to be more helpful. So at a higher level than specific level.
Makes sense. The other question is if the new species kind of also in Veracruz is green, do you think there's any maybe relation to the same pollinator or some interbreeding between maybe Calcicula and the other green species that could have created the green in this one?
Could be. The flowers in Beschorneria, ours starrii tends to be the most green of them all, but they all pretty much do have some green component to them. Calcicula is one of the few that doesn't actually, and it has red. Interestingly enough, I didn't mention this, but it's something we had to work through with in the paper. If you want to read it, it's out there. And Calcicula was actually described as having pink red flowers, occasionally yellow, which makes no sense, right? On the color scale, you know, how you get from a red to a yellow. Now, how you get to a green from a yellow, like we saw in flowers that were fading, easy, right? Yellow's a component of green, right? Yellow and blue makes green. But going from red to yellow just seemed very strange. So what we've postulated is likely when they were doing the Beschorneria analysis, because they incorporated populations from multiple populations that they didn't have, they thought were all Calcicula, and some of them clearly were starrii, that they may have had old specimens, old flowering specimens of starrii with that fading yellowish flower. And ironically, at one of the localities where we found that was reported as Calcicula, but we determined and found flowering specimens to be starrii, we had one inflorescence where the whole thing was perfectly green, all the flowers were green, one yellow flower, pure yellow flower. Very strange. So, but for starrii, like I said, not too unusual, right? Going from green to yellow, but going from red to yellow, that's that. So we think that probably the yellow reference in Beschorneria calcicula was probably starrii specimens at the time. So, But as far as the new taxon, which Bescherneria carolinae is the new one that was described that gave me a heart attack in December. I suppose it's possible, but like I said, there's a lot of green and a lot of different flowers. It just had the closest amount of green to what ours have, right? It's starry eye. The other character is extremely different. So pollinators and generically, I think you may be correct. I suspect, and I would love to do a long-term project on this, that starry eye has more diverse pollinators than Calcicula. Calcicula has, more concentrated on hummingbird pollination syndrome because of the red color. But starry eye perhaps has expanded a little bit more into the insect world and has moth pollinating going on, represented by the green flowers, but also attracts hummingbirds clearly because they do have this red stem, this pink stem. So it still would attract hummingbirds, similar to like my Fucaria boragi outside my door is white flowers, but it gets visited by hummingbirds as well. As far as intergradation of the two, I don't know if there's enough evidence at this point for that, simply because there is enough variation in flower color that green is not, out of scope for almost any of the Bescherneria. And all of the other characters of the plant were extremely different. Habitat, elevation, the very distinctive wide leaves. In the description of carolinae, they actually postulate that it's an intermediate between Bescherneria albiflora, which is this very large plant, often caulescent, so it has a stalk with very wide, giant leaves, and the tubiflora / Calcicula group as it was known before our paper was published. So there's some kind of speculation with not a whole lot of basis for it, but that's what science is for. Is speculating and determining whether you have the data to support it or not at a later time. So could be, but I don't think that plays a role in it, just because of the differences in habitat and geography historically in this area.
I was also just thinking Hesperalo. I think, isn't there a red and a yellow flowered form of Hesperalo?
That's
In
correct.
cultivation. Yes. In cultivation. Yes, absolutely. Yep.
But and then my last question is, could you just talk a little bit about the conservation? Because I know you, it sounds like you took a lot of former habitats for Calcicula off the map, either took them off the map or converted them to starry eye habitats. And what's the range kind of, or what's the conservation status of starry eye itself? So you could just talk a little bit about both those.
Well, what's interesting is the result of the study conservation wise kind of flipped the tables, because Calcicula previously had this very wide range, north to south, across two different mountain ranges on the other sides of the valley. But because of our research, most of that is starry eye. Calcicula is now very tightly restricted to the very south of the Victoria Valley. So even though starry eye is probably under no immediate threat, now it definitely is under threat at the type locality, because you could see in the background where mining was taking place, they're literally taking these isolated hills down piece by piece. So we don't know how long that mountain range, that little hill range will be around, because it's not extensive. And in the paper, you'll notice we did visit other isolated hills in the area, trying to see if there was, they were distributed elsewhere. There was one hill we identified that looked perfect for it, but we couldn't get permission in to see, to check out that hill because it was owned by a mining company. So starry eye overall probably isn't at a point where it's threatened, just something we need to keep an eye on, but it has enough localities, extensive localities, that I think it's in a decent spot right now. However, Calcicula, different story. Now it's got this very restricted range. We know at the type of locality, there's heavy pressure from goat grazing. And we know at other localities, that we suspect are Calcicula are either gone completely, or the habitat's changed naturally, because natural stuff happens too. That's why I say that pine forest, it looks like it just happened naturally, but 40 years of a growth of a pine tree, makes a big difference. And there were a couple localities where we did find plants, but we didn't have flowers. So they're presumed to be Calcicula. Not 100% because we didn't have flowers. So it turns out that Beschernera calcicula is actually more of concern conservation-wise than Starrii. So that's the kind of conclusion that we came to, is like Starrii may not be in immediate danger right now. We should still be monitoring because there's so much pressure in the area. But calcicula is actually a little bit of more concern because now it's very range restricted.
Right. Did you want to ask a question from the Q&A, Rod?
Yeah, I've got my own question too. First, are any of these 3 species in cultivation yet?
No. Well, I shouldn't say no. Calcicula has actually been in cultivation for a while. Matter of fact, one of the reasons why my brain went to Beschernera originally, when I got down from the mountain, I pulled up that Agavaceae.com immediately, was because I had actually, when I first moved to Arizona, I wanted to grow desert things, but not the things that I saw in my neighbor's yard. So my whole life is be different, right? So I always try to grow different stuff. So that's how I got into Fucaria, because I saw Fucaria splendens everywhere. Well, no, I didn't want to grow that one. I want to grow the other one. So I grow all the other 10 species of Fucaria in my yard. And early on, I came across Beschernera, and I knew it was an agave relative. Didn't seem to, not many people had it in cultivation, certainly not in the US. In Europe, it's actually, Beschernera is a fairly commonly kept plant. They have cultivars that are available from breeding and things like that. And as I mentioned in the paper, it's really hard to determine the genetics of those guys because it's been so many years and open pollination, and nobody's tracking data. So we don't know too much about the ones that are in cultivation in Europe. But in the US, I was able to procure about 6 species way back in the early 2000s when I first moved here, one of which was indeed calcicula. And I think I got them all on eBay at some point. And all of them promptly died on me, except for calcicula, which is not too unusual because it's one of the species from a more arid environment. It stayed with me for about 2 years before it finally gave up the ghost. And so I was somewhat familiar with the genus and sort of remembered calcicula. I pulled up my old photos of the plants that I had in ground and things like that. But since then, I haven't really found anything that's available for plants within the States. You can still get them from a couple of places, not calcicula though. However, if you're willing to grow from seed and you're willing to get a USDA import permit for the seed, which is very easy to do, a small lot of seed permit, RPS, Rare Palm Seeds out of Germany, does sell seeds of several species, including calcicula, and it has for years. However, keep in mind, what they call calcicula was what it was called when they went and did the collection, right? So were they collecting calcicula or were they collecting Starrii? We don't know, right? From what we've seen, I've actually ordered some calcicula and started to grow. From what it seems like they got it from calcicula locations. So they do seem to come in up as calcicula. So I do believe calcicula is in cultivation based upon RPS availability of seed. Most people don't grow it. Clearly not something you commonly see, but it is possible to obtain calcicula in cultivation. Have not seen any kind of reference to Starrii in cultivation at all at this point. So
We have one question from Q&A, and it's a pretty easy one. Yes, it's true that these plants, Beschernera is not monocarpic. It's like yuccas. It continues to grow and flower again after flowering once.
well, yes and no. And Greg and I have this ongoing discussion about, monocarpic versus not monocarpic, because what they do is they offset heavily, right? So even though an individual rosette may flower and then die, by the time it does that, it's old enough, it is actually offset to a cluster. So you'd see these very large groupings of Beschernera, and they probably are the same clone. So one rosette flowers and it dies. Does that make it monocarpet or polycarpet? Depending on your definition of it and how you view it in the overall sense, you could probably say either way, right?
Then there's a couple questions about your methods here. The first one is, did you use drones or anything to look for the plants? And the second one is, did you use any DNA analysis, or is that something that you'd want to do potentially to compare with maybe Calcicula or something or another species?
The second one I get all the time, so I'll approach that in a couple seconds here. But the first one, no drones, manpower only, leg power. That was it. That's how we did it. Binoculars and hiking through a lot of hectia and painful plants and very steep slopes for some of these guys. But yeah, no advanced electronics were involved in this at all by any shape of the imagination. Regarding molecular analyses, it's interesting. I get this all the time, but I think it's important to understand what a molecular analysis provides, what kind of data they can contribute to a new species description. Because in reality, what the genes are is simply another character, right? Just like flowers is a character, inflorescence is a character, leaf shape, plant shape. Those are all characters. That's what the DNA is going to tell you. It's going to give you a character set for that plant. That is worthless without a comparative material, right? So if I have a green flower, dandy. But without comparing it to Calcicula, which has a red flower, there's no data involved in there except green flower, right? So you can't designate a difference without a comparative material. So that means, and molecular analyses are not cheap. They're getting cheaper, but they're not cheap. Number one, they take an additional expertise beyond what I have in my experience. It takes a lab, and it takes even more money. And so it also takes an additional collaborator that can understand and has the software available to do the analyses for molecular data and to, run the plates and things like that. You can often combine it with other samples from other studies to reduce the cost, but there's still a significant cost to it. And I am a recent adjunct researcher with the Biodiversity Institute, but I'm a non-paid position. It's a research position only. So I don't have the facilities nor the money required to do a molecular analysis because you can't just do it for one species. It doesn't have any value unless you do it for beyond the species. And then let's say we did get Calcicula DNA. We ran our programs and the programs basically spit out a family tree, right, of the input. What you'd find with Bistroneria, Starrii, and Calcicula, I would probably guarantee would show you a dichotomous branch. So Calcicula and Starrii are each other's closest relatives. Kind of knew that already, right? So to really find out, okay, is Calcicula more closely related to Starrii than other Bistroneria? Well, now I've got to go get other Bistroneria, right? So now you're increasing the scope of the project even more. So really, you're not describing a new species now. You're actually doing an analysis of Bistroneria and the phylogenetic relationships of Bistroneria. So it's well beyond the scope of just describing a new species. Because even if the DNA came out and showed that Starrii and Calcicula came out identical, that doesn't indicate that this isn't a different species. It simply means that you've looked at the wrong area of the genome to make that determination, because something genetically is making them different, right? So that's why it's important to remember molecular information is primarily useful at a much higher taxonomic level than a species level. Now, different story with something like Aethelocactus tepelmemensis. For those of you who don't remember, we didn't know what genus to put it in. It was so different from every extant member of Thelo. We were thinking Pherocactus, Turbinocarpus was even something was brought up at some point. Stenocactus, that actually would have helped. We'd have to have molecular analysis of a bunch of other genera, though, to help place where the Thelo was going to be, right? Which we didn't have the capacity to do, funding or otherwise. So we made it, whichever one it matched closest to, which was Thelocactus schmollii. So, like I said, molecular data is fantastic and is a great tool. but it is not a useful tool typically for a description of a new species. It's for a higher level of taxonomy. Does that make sense?
Yeah, that was perfect.